So I think everybody's aware that a little over a month ago we had the JRE podcast with Flint Dibble and Graham Hancock. About 4 hours worth of video there. Pretty interesting. One of the things that has occurred since then is a gentleman by the name of DeDunking. His first name is Dan, and Dan has taken it upon himself to be the White Knight for the Atlantean Mafia. Um, he makes some claims in a response video that I just thought was hilarious when I first heard it. Considering that he has 100,000 views, where he's literally misinformed people about archaeology, even though he claims he's some kind of great researcher. I just wanted to highlight the fact again, 100,000 people that he lied to, and we'll get into that now. So basically, what we have here, title of the presentation, and I apologize for the low tech um, response here. Look, I'm not getting paid for this, but 100,000 people being lied to, somebody's got to say something. So the title of this presentation, The Evidence Ignored. We're going to look at archaeobotany, paleobotany, bioarchaeology, and paleoethnobotany. Maybe some of them, all of them, none of them, but we'll get to it. So here's a clip from the Joe Rogan Experience, and essentially this is going to highlight one of the issues or one of the concerns that DeDunking had. Let's go ahead and listen to that.
Is it possible that this cycle of domesticating wheat and beans and all these different things has taken place many, many times? And that if you left them alone, they would go back to the wild form? How long would it take for them to revert back to their original
thousands
state?
Of years again.
Thousands of years again.
Thousands of years.
How many thousands of years you think?
Well, I don't know because I'd have to look that up.
Okay, let's go on to the next slide and I'll explain a little bit about what's actually going on there.
Is it possible?
So what we've got here is we've got a question from Joe, and Joe's coming at this from the angle of a lost civilization, and he specifically asks about wheat and beans. You can clearly hear Dibble correct his answer, because depending on how you ask this question gets you different answers from different archaeologists. And in particular, you need to ask if we're looking at genetic data, macro data, microscopic data, if we're looking at chemical residues, CT type evidence. So there's just this long list of stuff that you specifically need to ask for. So although I can see what's happening in the question and how Dibble wants to answer, technically, Flint's not wrong. The evidence for plant domestication is thousands upon thousands of years. There's no changing that, and it's based upon physical evidence. But we'll read this slide too, and we'll get to that point. So debunking follows this video or this question up with a reaction video, and he proclaims that wild rice and rice domestication cannot be tracked or understood based upon some genetic papers he has read. Supposedly, in these papers, in his opinion, refuting any ability to track domestication or start and stop domestication events, i.e., specifically here we are talking about lost civilizations. They have farms, they disappear. Would we ever be able to tell without them present or any record if they were farmers? Um, Dan, while reacting to an archaeological conversation, however, skips physical archaeological evidence and instead lies to his audience using non relevant genetic rice studies. Dan has been recently on X, and he's been touting his research skills, and he says the vast amount of scientific evidence I brought to bear, and he's directly referring to a comment we'll show you here in a section. In a section, in a second, the vast amount of scientific evidence brought to bear and he's recently posted this on X, and we'll get to that here in just a second. But I just wanted to point out that pseudoscience consists of statements, beliefs, or practices that claim to be both scientific and factual, but are incompatible with the scientific method. Meaning, you can stand in front of people and bring in scientific papers and read them, and if they have nothing to actually do with the conversation at hand, or if it's the wrong type of evidence for the conversation at hand, then we need to set those aside and look at other lines of evidence. It's kind of clear from this discussion that people don't know what archaeologists actually do, nor how archaeologists actually actuate various lines of scientific investigation to get to the conclusions that they get to. So let's go forward. So this is the portion that I had a hard time with because I just saw it as intentional. It's very deceitful at best. Best, it's just ignorance. But again, I've seen Dan talk. I've seen his videos. I don't for 1 second believe that he did not do this intentionally. And I'll explain. But first, we're going to listen to what he has to say.
And he merely told Joe he doesn't know how long it takes. And this is not a small thing. Joe asked a very pertinent question, and Flint shut it down with a confident statement that excluded any possibility of Joe's idea being true. The statement, thousands of years, is false. In 100 years, it is clear in the record, and by 1,000 years, you may well not be able to tell if it was ever domesticated at all, as we see in the record of rice today. Flint, he merely told Joe.
And so what you just heard there is 100% not true. 100% not true. For several plant species, you know, we can track when they're wild, when they're domestic, when they go to weedy. We can separate out different variants of those things. So I'm not sure how much research Dan actually did on this to think that he was qualified to make the comment that he did, but it appears very little. I can understand perhaps his confusion about Dibble's answer, just like I'm concerned that Dibble didn't quite understand what Joe was asking, or Joe didn't quite understand how to ask the question appropriately. To ask Joe's question appropriately is actually a multi-parter. You would need to say, hey, what evidence through time do we have of domesticated wheat, rice, bananas, palms, geophytes? And you got to go down the list and say, okay, for these, we've got really good and strong information for that maintains integrity in the archaeological record. For these, sorry, we don't have anything. For these, we're working on. But we didn't get that interplay here because archaeological evidence was skipped. We just have an individual that picked one particular topic that seems, well, it seemed to him to get some sort of dunk on somebody, and it's just not true. So in short, the goalpost was moved, and an illegitimate information is presented to the watcher or listener. We call this a lie or lying by omission. Dan would refer to it as a red herring. And oh my God, this is a big red herring. So how do we fix this? How might an archaeologist document prehistoric cultivation, domestication, and/or start-stop domestication events? Well, let's get into that. This is one of the lines of evidence that Dan didn't want to talk about, but I think Flint was referring to. Flint's, he's thinking of macro evidence, the seeds you collect on site, and how long those last in the soil, and all the other evidence that we're pretty accustomed to reading about. But let's go further. So phytoliths. What we're going to talk about here is phytoliths. Phytolith just means it's a plant rock. These are diagnostic plant microfossils. You can also throw calcium oxalate in there as well, maybe some other things. Phytoliths have been known for about 200 years. They were discovered by Struve back in 1835, and these little guys have been used in archaeology perhaps as long as 70 years. Been a little bit lazy. I didn't go look that up, but it's a long time. Phytoliths, what are they? Phytoliths are biogenetic silica, opal phytoliths, people call them plant opal. These are the internal biological silicon structures of plants. Essentially, silicic acid is absorbed from the ground by plants and deposited internally into and between various cells. Once in the plant, the silica is converted to silica dioxide, SiO2, essentially becoming a casting of the cell or intercellular space, resulting in a potentially diagnostic shape. Um, I haven't looked this up to see what the current list is, but at one point in time, there was a list out there. You can actually date the phytoliths. Yeah. So here we go. So we have a plate here of some phytolith micrographs. If you wanted to use these pictures, just go ahead and cite this video in the future. But we can see here photograph A. These are this is a phytolith from a ponderosa pine needle. B is also a phytolith from a ponderosa pine needle. You can see the similarities in body structure. One is just longer than the other. C is a granular, circular, bowl-shaped phytolith from the ponderosa pine seed. D is also shaped, granular, bowl-shaped phytolith from ponderosa pine seed. The difference being that D is intact, where C has been broken up a little bit. E, let's go to letter E here. E is a spherical globular phytolith from ponderosa pine cone bract. Looks a little bit like um, pine pollen, but I can guarantee you it is not pine pollen. That is silica, blacks out under polarized light, other various tests. Anyway, F is a silicified segmented trichome hair from arrowleaf balsam root. G, this is actually a calcium oxalate. I threw this in here because we mentioned it. It's a calcium oxalate raphid needle from Chamasia quamash. And some of you guys might know that's pretty popular North American plant. And this was broken up out of a much larger needle bundle from that plant. So if you guys know anything about that plant, you probably were just surprised to see that guy as I am. Anyway, H, lodgepole pine needle phytoliths. And so the issue here with presenting these is just to start showing you the microscopic level of evidence that we get from plant remains. When plants are processed or they deteriorate in the ground, the soft tissue goes away, and we're not left with those organic bits and pieces. What we're left with are these inorganic bits, these castings of various cells or cell structures. And these last in the soil for a long time. As long as the soil is hospitable, these guys are going to be there. They're stuck to tools. They're in the soils. You can find them on mammalian teeth. They're all over. You can get these out of your lake and bog sediment cores. You can get them out of cores from the ocean. And it's a good way to go back through and recreate um, your plant taxa environment. Anyway, let's go to the next one. So wheat. Wheat was specifically one of the food crops brought up by Joe. In the video, Flint does give him credit and say, okay, yes, you are correct. This guy, we know how to track this guy from the macroscopic level and the genetic level. Unfortunately, this is where we kind of, jump the shark a little bit here, because Dibble, nowhere in his response was he actually talking about genetics that I could see, at least from the transcript. Although it's kind of mentioned, I think it's inferred in the conversation. But in general, Rogan's asking about what evidence can we track in the archaeological record that would get us solid answers potentially about domestication, start-stop domestication, evidence for a lost civilization. So here we are. Off to the right here are pictures of wheat phytoliths, various of those. Some are diagnostic, some are not. But the important point to remember here is that, as it says here, wild domesticated wheat and barley phytoliths can be distinguished from each other at the genus level and from common weed genera, which is to be expected in archaeological contexts in certain regions of Southwest Asia. And we're going to focus on Asia here for a reason because, mm, you know, rice was mentioned by DeDunking. I don't know why he brought in rice DNA because it gets you nowhere for this purpose. More work is needed with other wild taxa outside of Triticum and Hordeum to more broadly apply phytolith identification schemes when congeneric non-cultigens may be present. Certain kinds of domesticated wheats can currently be distinguished from others and from barley using specific types of phytoliths or combinations thereof. See Persol, Lentfer, and Zhang, 2015. This is where this information came from. And I wholesale took these photos from them as well. Hopefully they understand the importance of this presentation in this big lie, and I can get away with that. I won't get any nasty emails. Next slide. So here we come to rice. Rice. This is so this is the physical evidence that an archaeologist would look for working in an area where we think wild rice, domesticated rice, weedy rice would be part of the human interaction on the landscape, right? And so, to date, bulliform phytoliths with 9 scale-like decorations are widely used signatures of domestication. We've known this since 2002. Over 20 years, we can identify domestic rice in the record, and these phytoliths aren't going anywhere. They're there. This is definitive evidence, one way or the other, to distinguish between what's going on. Not DNA. DNA is not how we answer archaeological questions. We need the physical evidence. So we have an entire plate over here on the right, various domestic, non-domestic types. We really won't get into that at all. But the pictures are from Wang, 2019, Plant Science, Volume 10. Next slide. And just to hammer this home, that it's important if you're going to do research and talk to 100,000 people that you actually do research. Don't find papers that support your bias and your suppositions and your desire to defend somebody else. I can get why you want to defend somebody else, but we can't tell lies doing that. We're grown men. We have kids. This is not the kind of example we want to set for the world, and we particularly don't want to lie to curious folk about what we can and can't tell from an archaeology side. So I've posted in these paragraphs here just to quickly highlight some of the information that's here that goes against the claim of DeDunking that we can't tell rice domestication. So to date, 3 distinct phytolith morphotypes have been identified, so that's important. Double-peaked gloom cell phytoliths are unique to the genus Oryza and can separate domestic rice from the 9 wild rice species. Okay, again, we've known about this since 1995. I bet it goes back further than that. So here's Dan telling us, oh man, we can't tell if rice is wild, domestic, weedy, disappears, come back. We can always tell because it's going to be in the stratigraphic column of soils. It's going to be on the tools. It's always going to be there. Unless preservation really sucks and something has destroyed these microfossils. But for the most part, they're there. And that's probably important to mention. If they're not there, then we need to look for other lines of evidence. Perhaps we need to bring in pollen. Perhaps we need to look at starch. Perhaps we need to look at chemical analysis. It's always something to go after to try and help figure out what's going on. But let's continue. In the past 2 decades, morphological features, including surface ornamentations, have been employed to distinguish domestic from wild rice. Old news, old news. And this is part of the problem. This is why it was laughable watching Dan's video. This information is so old. This isn't new technology. And for whatever reason, he couldn't find it. And I think it's because he didn't want to find it. He wanted to go after Flint and make Flint look bad and stick up for Graham Hancock. And it's bullshit. Let's continue. Phytoliths can also be used as a tool for understanding the development and spread of rice. So yeah, you can track this stuff across landscapes. It's pretty cool. We won't discuss that there, but look at the papers that are mentioned. Morphotypes from different field types reflect their arable system. When applied to archaeological samples, the results demonstrate changing farming practice over time. So not only can we tell wild, domestic, weedy, um, non-breakable, breakable, not the correct term, shattering, non-shattering. But we can also tell when the rice field is wet rice versus dry rice farming. So lots of detail in there, lots of information. I don't know why Dan told you he did, but it's not true. Off to the right, we can see the double peak gloom phytoliths from Zhao, 1995. Interestingly, in 2020, 2021, I think this was published finally in 2022, there's actually a new form of phytolith microfossil that they found. It's the fusion echinate phytolith. And this guy seems, if it proves true, will really help us push the envelope of what we can and can't identify that's important in an archaeology site. Because this guy here might actually reflect the non-shattering phenotype of rice within the AA genome. Our study adds detailed information regarding phytolith morphology and distribution, provides new tools and insights for the future of archaeobotanical research. GE 2022, phytoliths and spikelets have selected rice species from in situ observations. So that's pretty cool. Even if you had the macro remains and you said, this looks like a shattering version of rice, so these guys weren't farmers. Well, technically, what you can do is you can start looking for this type of phytolith. Hypothetically, at this point, we're still waiting to prove this out, I think. But if you can find this phytolith in your macro remains, it's going to actually push you over from that gross macroscopic observation to the microscopic and go, no, this is actually non-shattering. Anyway, pretty cool. 22 evidence that Dan didn't want to talk about. Okay, so the big overview. It's one thing to talk about phytoliths, talk about their existence, what we can and can't do with them. Has it actually been actuated in the field? Have we actually put this line of evidence with archaeological assemblages, and has this been done region-wide anywhere? So that we can understand cultivation, domestication, and maybe going back to the wild. So what we're going to use here is Yan Pan's 2023 article, revisiting the archaeological investigations of rice domestication in China, 10,000 to 7,000 BP in a human behavioral context, which is pretty cool. Okay, So he breaks this down into 4 rules for using different levels of plant physical evidence to figure out what's going on. Not DNA studies, physical evidence, right? So here we go. Massive efforts have been invested in the study of rice domestication and have generated many methods of distinguishing wild and domesticated states of different forms of rice materials, laying a foundation for exploring the process of multiple layers. So here's the first one. One, the percentage of domestic type rice spikelet bases. Two, the proportion of bulliform phytoliths with greater than 9 fish scale decoration. The morphometrics of double-peaked tubercle phytolith is also sufficient, indicating domestication. And number 4, to some extent, seed size may reflect the intensity of rice domestication. So, and this is important. So instead of taking a paper and going, oh, there's no domestication, physical evidence. Physical evidence. What is the actual record that's being, you know, taken from these sites and now investigation? We have 4 basic rules here, and that was used to guide his next few steps here of looking at rice cultivation regionally in China. So here we go. The Middle Yangtze region, a transition from wild rice utilization to rice cultivation, starting by about 11,000 years ago and lasting till 7,000. No macro plant remains, right? No macro plant rice remains dated to around 10,000 BP have been uncovered so far. So it's all micro scale stuff, micro evidence. The thousands of years long rice cultivation was demonstrated by a continuous increase in proportions of domestic double-peaked phytoliths. So it wasn't demonstrated with DNA studies. It was demonstrated with physical evidence studies. Phytoliths to be an example of this particular case. Rice cultivation is associated with groundwater table fluctuations 8,000 years ago is evidenced by double-peaked phytoliths and pollen data. So we have two levels of scientific investigation here, 2 different types of microfossils. From 9,000 to 8,000 BP, 530 kilometers away in the west, are the subsequent important sites. I'm not even going to try and say that name. The rice remains are husked and chaff tempered in pottery sherds. So there's another level right there. So you have pottery sherds, and you're finding these microfossil. They're embedded into the clay, or at least the impressions are, right? Maybe the whole piece, but they're pulling them out. So the phytoliths are surviving at high temperature, firing these reductive atmospheres for pots. Lower Yangtze region. The Yangtze region has yielded the richest materials for documenting early rice domestication. Phytolith analysis of the profile of probable ditch structure showed a relatively high density of double-peaked tubercle phytoliths with a few bulliform phytoliths dating 9,000 years ago. An abundance of phytolith analysis has also evidenced rice domestication, in particular, clarifying rice cultivation during 10,000 to 8,000 BP as a gradual process of rice domestication throughout the Neolithic. A gradual process of rice domestication throughout the Neolithic period was demonstrated by phytolith data, not DNA data, physical evidence. It indicated that rice domestication was initiated in Shanghai culture, and its intensity approaching the level of modern rice agriculture took place after the late Yabang culture, approximately dated 6,500 years ago. The Upper Hua River region. Analysis of rice spikelet-based morphology at Balangong reflected an established domesticated rice population had already existed In pre-Yang Shao period was sustained throughout the entire Neolithic and Bronze Age. Phytolith investigation suggested that the wetland rice cultivation was substantially practiced and anthropogenic water conditions changed over time. So again, that particular one, I found that one the most interesting because they had rice cultivation, and you could see the weather change in the physical evidence of these microfossils and detect that. High wet years, low wet years. Anyway, rice cultivation at Zhuzhai and Tanghu, sorry guys, as early as 7,800 BP was documented by the morphometric analysis of phytoliths. Next region, the Lower Hua River region, 8,500 years ago. Phytolith analysis played a significant role in verifying rice agricultural practices. While most sites yielded only a few carbonized rice remains, they did find paddy-like features revealed at the site, evidenced by phytolith data. It might have been the earliest rice paddy that we know of to date, going back 8,400 years. This means that rice cultivated in the region was relatively mature when it had just appeared. So that's that. You have one area where folks kind of attached early on to this practice, and in other areas when they adopted it, they already knew what to do because they watched it from these guys, brought it from these guys. People from here moved to there. You get the point. The domestic type of double peak phytolith shows a more remarkable rising tendency, but with more fluctuations. Next region, the Min River Valley. Typical rice phytoliths were uncovered from pottery sherds again. The cultural mid in the barrels, the PBS value of bullform phytoliths rose from 44% in the DeDunkings period to 72% in the Tianshan and Huangshan, sorry guys, periods, implying a seemingly intensified process of rice cultivation. So again, in the Min River Valley, low cultivation increasing through time. Pretty cool. The quick overview for all of this, though, is just this. 10,000 to 7,000 years ago, surprisingly, we can discover that rice rapidly dispersed across a vast area, closely following the threshold of its being cultivated by humans, while the rate of rice domestication early stage was rather slow. The middle and lower Yangtze regions, parallelly regarded as the original places of rice domestication, are supported by not only empirical evidence, physical evidence in your hand, not DNA studies, but also regression modeling. Cool stuff. Many details are yet to be discovered, and proposed diffusion routes need to be tested. Pan 2023. So let's listen one more time to Dan's claim, and regardless of the muddled nature of what he's attacking Flint for, I don't really care about that. Um, it wasn't a debate; it was a conversation. You've got Graham Hancock in the room with Joe Rogan, so you got two guys against you. When he says it's many thousands of years, he's correct. He's 100% correct. He's not wrong. Maybe it didn't come out correctly, but DeDunking sure as hell didn't get it right. He was, everybody else is in there playing ball. Dan's out in the parking lot. He can't even get out of his car. He's just, he's out there. Anyway, let's listen to this one more time.
And he merely told Joe he doesn't know how long it takes. And this is not a small thing. Joe asked a very pertinent question, and Flint shut it down with a confident statement that explained he merely told Joe he doesn't know how long it takes. And this is not a small thing. Joe asked a very pertinent question, and Flint shut it down with a...
Yeah, so, and let's see this.
And he merely told Joe he doesn't know how long it takes. And this is not a small.
Yeah, so he didn't. His, his answer is correct. You know, answering back that I don't know how long it takes or how long it will last, or expressly what evidence I need to present here. But we've all been reading these papers for over, for 30 years now. We know that they're trackable, we know that they're traceable, and we know that they last. Um, so to see him stumble a little bit on that, I'm not concerned about that because it doesn't matter how you come back at this question, he's going to be right. He's going to be right. You already said he was right on everything else. You only said he was wrong in this one issue, which is, and he wasn't even wrong in this issue until you brought in rice DNA. Then he's wrong because he didn't say anything about rice DNA, did he? No, he didn't. You introduced rice DNA to try and pretend that he had said something wrong. Let's go ahead and finish listening to this if we can, if I can get it to cooperate.
Small thing. Joe asked a very pertinent question, and Flint shut it down with a confident statement that excluded any possibility of Joe's idea being true.
Yeah, because Joe's idea isn't true. We all know this, and whether or not Flint was prepared to go through all of this evidence, and it's long. It's long. Just like I said, you didn't talk about the wheat and the rice and all these other crops. You got to go one by one because it's not all encompassing. So Joe's question is not correct. He's not going to find the evidence that he was looking for his lost civilization here.
The statement, thousands of years, is false. In one
No, the statement of thousands of years is 100% correct. The evidence less multiple thousands of years, and you can pull up your DNA studies, but that's not going to help you understand the true physical evidence.
Flint, he merely told Joe he doesn't know how long it takes. In 100 years, it is clear in the record, and by 1,000 years, you may well not be able to tell if it was ever domesticated at all.
Yes, as soon as it's domesticated, we can tell DeDunking. We can definitely tell, and after 1,000 years, we can still tell, and several thousand more years. I don't care if they had domestic rice for 5 years and then stopped. Right? If you go in there and you can find that soil profile, that core from the lake, the pond, the ocean, that piece of pottery, phytoliths from the tools, wherever, we're always going to be able to tell. Always, always, always going to be able to tell, man. Physical evidence, buddy. This is I'm amazed. It just frustrates and amazes me that you went this route. Like I said, there's no way you could have missed this stuff. I think you did this intentionally, just to try and get one over on Flint, which kind of sucks, man. Like I said, we're men. We shouldn't be doing that. You got kids.
As we see in the record of rice today.
All right. So, and he finishes off. He says, as we see in the record of rice we see today, but he didn't look at the record of rice that we see today. He went with a DNA study, which was wrong. He selected the wrong evidence to analyze what was going on and what was germane to the original question. You know, Joe Rogan's question was, would we be able to see if somebody was domesticating crops and then those crops went away? Would we be able to go back in time and detect that? The answer is 100% yes. It is yes. It's always going to be yes, and it's going to last for multiple thousands of years. Let's go to the next slide. Lastly, let us invite Dan of DeDunking to argue with more PhDs with the following quote. DNA technology has been considered another exciting tool for detecting genetic data of rice domestication. Here, we refer to the application of DNA techniques, in particular to investigating archaeological remains. Unlike animal and human bones, plant remains do not yield DNA fragments that are good enough for polymerase, chain reaction, and sequencing. PAM 2023. So here's a 2023 paper that lets you know It lets you know DNA is probably not a good way to come about answering Joe's question. Yet DeDunking did it. He claims to be a good researcher and he's got this down and he's the man that's going to always find people not telling the truth. Well, what the happens when you're the guy not telling the truth, Dan? You've got 100,000 people that listen to your BS because you were too lazy to do research, yet you jumped online to tell people how Flint was wrong to the point where people are calling him at his job. You lied. He didn't. Joe's question was a good question, and he was given an appropriate answer. It's going to last for thousands of years. If you don't like it, call up Pan, call up other geneticists, ask them. The fact that your papers are older than 2023 should be a red flag to you. It really should be a red flag to you. The fact that you don't know what archaeology is or how we actuate various investigative tools to come to our conclusions is laughable that you're even talking about archaeology, Dan. I've watched other videos of yours, and you do this selective mistake a lot. In conclusion, Dan DeDunking likes to tell stories using pseudoscience tactics in order to defend Graham Hancock. He is 100% not an honest broker. He will lie to your face to win an argument. When Flint says the record for domestication lasts thousands of years, Flint's not wrong. Contrary to Dan's nonsense fantasy, if the soil conditions remain hospitable, you might say that the evidence for domestication lasts forever, even underwater. That's all we got, guys. Yeah, I shouldn't have to post videos like this, but DeDunking should shut his trap when he doesn't know what he's talking about. You know, one of his big hangups is the whole Sink Mars, um, Bluefish Caves thing. He makes a big deal out of that. He's like, "Oh, there were people in the room. They were making fun of Sink Mars." Well, technically, Dan, there were guys in the room in the back that didn't understand what was going on. They were just jabbering, um, talking about things they had no idea about, things they probably should have kept silent on and listened more. You're the guy in the back of the room, Dan. You really are. That's all I got. Thank you.